Welcome everyone once again for tuning in today. I'm very excited to present today's webinar, which is on essential tips for choosing and using mouse models in research. Before we begin and dig into today's topic, I would like to take a moment to introduce to you the Jackson Laboratory. The Jackson Laboratory, otherwise known as JAX, is an independent nonprofit biomedical institute. Our primary mission is to discover precise genomic solutions for disease and empower the global biomedical community in the shared quest to improve human health. Now, JAX is a global experience that translates directly into a global influence. We have 3 locations here in the United States, as well as an extended presence in China and Japan. In order to support our mission, we strive to empower scientific excellence in order to drive innovation. We drive scientific innovation in 3 main ways. One is through discovery. We have our own internal research core with their own funding that performs research that is beneficial for the scientific community. We next drive innovation through research. We perform or provide various mouse models to support research and scientific services to our customers. And lastly, we promote innovation through hosting a variety of education-related activities, such as providing opportunities for research, conferences, and hosting live and on-demand webinars like the one you're watching today. Today, we will cover methods and resources that can help you find and choose strains more quickly. We will also discuss the basics of strain nomenclature, which will be useful for identifying strains faster. We'll also go over strain background and why it matters, choosing controls and other considerations when maintaining a colony to help ensure that the data you generate with the mice will be reproducible over time. Some of these topics are covered in extensive detail in other presentations. However, my goal for today is to give you a broad sense of what you may want to think about when you are just starting out and planning your experiments. Today, we will start with one of the more difficult parts of planning your research. So how do you find and choose a mouse model? More often than not, when folks are first trying to search for strains in our repository, the first place they visit is our main page. When you visit our main page, which is jax.org, it'll look something like this. In order to search for strains from this page, go to JAX Mice and Services tab, here which I have highlighted in yellow. From here, hover over Find Mice and Biospecimens and open Search JAX Mice. When you click the Search JAX Mice tab or button, the page will look something like this. You can also access this page directly by using the web address listed below on this slide, which is mice.jax.org. Because the Jackson Laboratory has so many strains, I would like to discuss some good search tips on our website. This search is mainly for strains available at the Jackson Laboratory repository. However, there are more repositories available for you to search for additional models. Using the search box at the top, you can search by strain name, gene, allele information, strain description, disease, phenotype terms, research area, and even donating investigator. Use the autocomplete terms whenever possible, gene names or disease terms. And if a result does not come up, you can try synonyms as well. We also have curated lists of models for specific diseases, as well as some featured strains. On the left side, we have filters that you can use to narrow your searches. These filters are incredibly helpful for finding models. So let's take a closer look at some of the available filters. So, for example, we can narrow down by usage. Some strains are only available for nonprofit research. We can also narrow down by career-expressed, inducible knockouts, live mice, etc. So let's take a look at an example to see how we can find a mouse strain by using these filters. Let us look for an interleukin-2 knockout model. If I search for IL-2, which is the shorthand name for this interleukin-2 gene, you will see that I'll get 80 results. Since we are interested in a knockout of interleukin-2, we can filter these results for any null or knockout mutation. If we select null knockout from the left side of the strain attributes box, we are able to narrow down our results to 60 strains. Now, another commonly questioned asked by our customers is learning whether or not the strain they are interested in is available. In other words, is it live on the shelf? In order to figure this out, we can narrow down this search by selecting the live mouse filter, which is located under the products box. Now, you should see that you have 17 results that fit your criteria, which again was a live IL-2 knockout strain. And now all that's left for you to do is decide which strain is the best fit for your research. Your applied filters will show up at the top of the filters section, and you can remove them by selecting clear all, or you can individually remove them as well. Going back to our handy filters, there are a few others I would like to emphasize. JAX has thousands of CRE strains, and finding the right one for your research is important. If you're looking for something CRE related, such as CRE expressing or a CRE reporter strain, then these filters are your best friend. So let's look at an example together. Let's say we're interested in searching for a liver-specific CRE strain that is on a C57 Black 6 background. I would start by searching the key terms liver and C57 Black 6. Then, from the filter section of this page, select CRE expressing in the platform section and live mouse under products. You will see that when you apply these filters that you'll get 15 results for this criteria. Again, all that's left for you to do is choose the model best suited for you. In addition to this CRE filter, we also have filters for conditional ready mice, such as flocks, or inducible models, such as tamoxifen-inducible ones. Again, once you have found a list of potential models, you'll want to review information about the strain to see if it would be a good fit. So how can you find the strain data sheet? You can do this by selecting any of the strain's names, which is in blue. For today's example, let's say we're interested in the fourth mouse listed from our search. The strain data sheet is going to give you a ton of information about the strain. The first section is the overview section, and it will contain information such as the strain's full name, the strain number, and a general outline of the strain. Next to overview, we will look at the remaining sections briefly, starting with the how it's made section. This section contains 4 parts. These include the strain highlights, a detailed description of the strain, the development of the strain, and more information about the genetics of the strain. So let's look at each part a little bit more closely now. The strain highlights will give you information about the background, type of mutation, the donating investigator for the strain, and has links to references as well. Continuing down the data sheet, this detailed description is going to give a more in-depth overview about the strain. Carefully reading the section will give you a good idea about whether the strain is the right one for your research area as well. Then the development section will give you information about how the donating investigator developed the strain, including the construct information and what is known about the strain's history. Finally, we have the genetics section. Here you'll find information about the generation that the colony is currently in. If you're unfamiliar with generational notation, there is a link here in this section that will provide definitions and explain things a little bit more thoroughly as well. The allele box below will also provide you with specific information about the mutation itself, such as the location of the mutation. And for this example, we see that this one is on chromosome 13. Next, we will view the how it's used section. In this section, you will commonly find selected references for the strain, which generally include the donating investigator's paper and any other related references that have cited using the strain. Additionally, if there is phenotypic data available for the strain you are interested in, that information will also be available in this section. Here's an example of what that might look like. This is phenotypic information that is available for the C57 Black 6 strain mice. Under husbandry, you will find a lot of information about maintaining a colony, such as genotyping assays, suggested breeding schemes, and suggested controls. This section is really great to look through once you have already narrowed down your search and are ready to consider purchasing a mouse. Since today we are focusing on model selection and how it's used, how they're used in research, I'll let you explore this section after the webinar more freely. Our final section of the strain data sheet deals with usage information. Some of our strains require licenses to use and can only be used for not-for-profit entities. It is always a good idea to check if there are any restrictions on the usage of the model that you're interested in. Now, let's say if you're searching for a model with a mutation, such as a knockout for a specific gene, and you're not finding any results through our JAX mouse search, it may be a good idea to verify the gene symbol that you are using. Next, we will discuss how to use the Mouse Genome Informatics database to find gene symbols and available mouse strains as well. Using our JAX mice search is great if you are familiar with the genes and nomenclature that you're interested in. Sometimes, however, it can be difficult to locate strains if you do not know the shorthand gene symbol information. In our earlier example, I knew the shorthand notation for interleukin-2, which was IL-2. But what if you don't know? How can you find the gene symbol? The Mouse Genome Informatics database is a great resource to look for this. So if a customer asks me to help find them a model for a gene that even I'm unfamiliar with, this is where I typically start. Not only will this database give information about the gene, but it's also a great resource for mutation information, strain location, phenotype data, and additional references. A good place to start is the keyword search for a gene or disease in the quick search box, but I also note that MGI has additional search-specific pages as well, which are shown here on the slide for you to explore. In this example, I am searching for a strain that has a knockout mutation in the AID gene. Let's say I've tried searching other databases and my search did not provide me with any answers. So I'm going to start by typing AID into the keyword search. This is what the search results will look like. You'll get information for allele symbols, allele names, locations, synonyms, type of allele, and some measurements of best matches. I have done some creative editing on the next slide so that you can see the information more clearly, and I highlight the parts that are most helpful here. First, we see the full gene name from our search, and we can verify that this result is correct because we also see that on the right side, AID is listed as a synonym. Even though we see that AID is a symbol that could be used to search for our gene of interest, it's not the official gene symbol. An alternative gene symbol we could use in the future while searching is AICDA. We can also click on the official symbol name that is in blue to get more information for that gene and use this also to search for strains. If we click the gene name, it will open a new page. This page has so much information, and I highly, highly recommend that you spend some time exploring the type of information that you can find on this page. For the purposes of this webinar, I would like to focus your attention on the mutations, alleles, and phenotypes box. On the right side of this box, there is a link to all mutations and alleles that have been created for this gene. What opens up is a list of mutations that have been produced in this gene. This one has 31 results, but we are only going to focus on just a few. The allele name is listed on the left, and the type of mutation is listed under the category. But again, let's say we're looking specifically for AICDA knockout model. In the shortened list, we see that there are 3 endonuclease-mediated AICDA knockout mutations. So let's go ahead and open one of the allele-specific pages by clicking on the allele name. This will open the allele-specific information page. This page has lots of information about that specific mutation, including mutation details shown by the arrow and enlarged at the bottom for you to view. The amount of information that is available for a mutation will vary based on a few factors, including how well characterized the mutation has been in the literature. This is also the one place you can find some Cree expression information for many Cree strains as well. The Find Mice row will also indicate if there are strains commercially available that carry that specific mutation, as well as indicate how many may be available carrying the mutation of interest. I have enlarged that section at the bottom for better viewing. In this section, if we click the available mouse strains link, which is signaled by the yellow arrow, you will be taken to the IMSR, the International Mouse Strain Resource. The IMSR is a multi-institutional, international, collaborative effort to provide a web searchable catalog of mouse strains and cell resources. This public repository search will find strains carrying your mutation of interest, associated synonyms, and the strain's current available status, such as is it available as sperm or live on the shelf, and link you to the repository currently holding each strain. I do want to emphasize that this strain example is held at the Jackson Laboratory, but this search engine does include other repositories as well. Of note, however, is that it will not list private strains or colonies. While you are searching for your potential research models, you'll most likely notice that mice have a very specific nomenclature. Although initially it may look daunting, it contains information that will help you determine the strain background and other important information. In 2001, the International Committee of Standard Genetic Nomenclature for Mice established guidelines for naming mouse strains. The nomenclature rules for mutant strains was put into place to improve reproducibility. Essentially, to improve reproducibility, the mice in a publication need to be specific enough for other researchers to find it. As I showed previously, there are many AICDA knockout strains. Each of these strains is unique from one another, which can affect the phenotype of the strain. MGI has a lot of information about these nomenclature guidelines, and this page gives some quick guides and more detailed information about nomenclature than this webinar will get into. So it's a great resource for nomenclature questions that you may have moving forward. JAX also has a handy guide that outlines strain nomenclature rules. A strain name will tell you a lot about a mouse, including the background, the type of mutation, the location of the mutation, and substrain information. So let's take a closer look at some different types of nomenclature. When reading strain nomenclature, the easiest first step is to identify the hyphen, which separates the strain background and the mutation information of the strain. In this example, going from left to right, the strain background is shown in black and is C57 Black 6J. To the right of this in purple, you will see that the gene the mutation occurs in is APC. In superscript, there is allele information. In this case, the mutation is indicated as MIN, M-I-N. And lastly, in blue is where the strain is maintained. Typically, the lab code is indicated to the right of the slash. In our second example, the strain background is B6, which is a shorthand for C57 Black 6. If a mutation originally was on a different background, then brought back over to another, this is called backcrossing. As such, the nomenclature will list the current background first, which here is shown in black, and the original background shown after, which is here in orange. Here we see a semicolon separating the two backgrounds, which indicates the strain has a mixed background of C57 Black 6 and 129P. Again, in this example, the gene with the mutation is in purple, TCRB, and the mutation has the letters TM, which means it's a targeted mutation. You may also see EM, endonuclease mediated, as well. The serial number indicates that this was the first targeted mutation created to the TCRB gene made by the specific lab code, who's in this case mom. You may see a variety of letters for lab codes, just so you know. And then finally, at the end in blue is our lab code, in other words, the repository maintaining the strain. As before, background information precedes the hyphen. I mentioned in the previous slide that the semicolon indicated a mixed background. Here you see a period. The current background, or period, there we go, which again, the current background is in black, followed by a CG, which indicates that there were multiple strains involved in the original strain. Technically, CG stands for complex genetics, but it is a good way to remember congenic. A congenic strain is a strain that has been backcrossed to a new background more than 5 generations. When backcrossed for 10 generations, the strain is about 99% identical to the new inbred strain. Additionally, because the strain is congenic, it will carry a period, whereas when it's on a mixed background, it will carry the semicolon, as we saw in the last example. Following background is the dash and the letter TG to indicate the mutation is a transgene. Then inside the parentheses indicates the promoter and the gene involved. In this example, the promoter is PDGFB and the gene is APP. After the parentheses, you see the founder line. Now, the founder line is important. When transgenic lines are produced, usually the construct is microinjected into the embryos, and the resulting mice have different areas where the transgene has integrated into the genome. So these different mice or lines may have differences in expression. So it's important to remember that for reproducibility, that you need to know which founder line you are working with. Next, we have the creator lab code, and then once again, the lab holding code. I briefly touched upon the differences between congenic and mixed backgrounds, but let's take a closer look into nomenclature for these two strains. There are times when you may come across two strains, like the one shown here, that have virtually identical nomenclature. In this case, both strains listed here have a RAG1 knockout mutation. The difference between these two strains is contained in that tiny punctuation symbol. In the top strain, you see a semicolon. This indicates that it was backcrossed less than 5 generations to the recipient background, in this case, C57 Black 6. The bottom strain, we see a period, which means the backcross to C57 Black 6 has been at least 5 or more generations. At 5 backcrosses, the strain is about 95% identical to that new background, in this case, the B6, and is considered something incipient congenic. At 10 backcrosses, it's considered fully congenic. The advantage of using strains that are congenic is that the genetic variability in the strain is reduced, and it can help to limit phenotypic variability that can confound experimental interpretation. However, some strains no longer exhibit an expected phenotype once they're moved from a mixed background to a congenic one. So you may still want to choose a mixed background for your research. While you may think it takes several rounds of breeding to influence the phenotype of your mice, it can take as little as 1 generation to alter the phenotype of your mutant. As you change the genetic background, you can expect it to change the phenotype. Now, up to this point, I've covered a couple of examples with the text in color, but let's walk through a couple more examples without the color to get you more confident into reading nomenclature. In the top, we see the background strain is BALB-C, and the gene of interest is PHEX, P-H-E-X. The allele is Hajduk, and the strain is maintained in the Jackson laboratory. In the second example, we see that the strain has been backcrossed at least 5 times to the D2 background and was previously on a C57 Black 610 background. The gene of interest is DMD, and the allele is MDX. Again, the lab code is J, indicating the strain is maintained at the Jackson laboratory. In our third example, we see a semicolon, which indicates that the strain is a mix between FEB and 129S6. The gene of interest is SNCA, and we see that this was a targeted mutation due to CMTM. The serial number we see for this strain is 1, which indicates it's the first targeted mutation for SNCA that was done. The lab code was NBM and indicates who did this targeted mutation. And J indicates the lab currently maintaining the strain, which in this case is the Jackson laboratory. Now that we've covered the importance of understanding strain nomenclature, I would like to take the opportunity to ask you all a quick question. True or false, can you determine if the gene of interest is a knock-in or knockout based solely on the nomenclature? So for those of you who said false, that's awesome. For those of you who may have responded true, the reason the answer is false is because nomenclature alone will not give you this information. Nomenclature is helpful in providing information on what the mutations the strain carries and on what allele, substrain information, but information regarding if it's a knockout or a knock-in mutation specifically, you'll need to dig a little deeper and read through the strain description or development for that strain in order to obtain that information. One last database I'd like to highlight briefly is the mouse phenome database, otherwise known as MPD. This is a great place to find published data related to a strain. Let's say we were interested in learning more about memory tumor latency. When we enter this in the search engine, we see that there is some data published that is related to this search. For this example, we will use the data phenotype strain survey measures as an example. If we click this, it will show us the following study, which we see contains memory tumor latency data from days after birth. Now, if we click this link, we see data available from this study, which was actually from Lifstead et al. Very briefly, in this study, they performed a strain survey to identify mouse strains that harbored dominant genetic modifiers that affect memory tumor latency. Highlighted in blue, you see the inbred background of FUBN that contains a transgene that causes mice to develop multifocal memory tumors at approximately 59.1 days. These mice were then crossed to different inbred background strains to produce 27 different F1 hybrids, which are the remaining mice listed on the x-axis, and the tumor latency period was measured. The main takeaway from this data is that a single backcross can result in a dramatic shift in phenotype. In some backcrosses, we see that memory tumor latency increases, while in others, it decreases. As you consider your next experimental design, take some time to consider what background you want the mutation to be on, or perhaps if there are inbred strains which may have specific characteristics that you are looking for. The mouse phenome database contains a variety of data, including information on lifespan, reproductive performance, blood glucose, body weight, etc., for many inbred strains. And that is data that is provided by investigators from around the world. This data can be an excellent source for choosing potential backgrounds or verifying baseline phenotypic characteristics of different inbred strains. It's hard not to emphasize this enough, but it's super important to understand your strain's background. Here are a couple of questions that could help you gather information and decide the choice of strain you wish to work with. For example, what strain was used to develop the stock? What strains have been introduced through breeding? What is the current breeding scheme? The current generation? Has it been cryopreserved? Has the genetic background been verified? Frequently, strain data sheets will provide this information so that you are able to make an informed decision about the strain. However, if you're getting mice elsewhere, you will have to try to rely on information that is provided by the donating investigator on the status of the mice that you're planning to work with, who may or may not be able to provide all of this information, or you may have to rely on a primary reference for the strain. All of these factors are important when designing experiments, drawing conclusions, and ultimately your credibility, because if you're not careful, the ability of other researchers to be able to reproduce your results may be affected. Once you have determined what model is best suited for your research, you will need to determine what the best controls are. Typically, researchers choose a control that is as genetically close to the experimental model as possible, such as choosing the inbred background. However, your experimental design will heavily influence your control choice. If your experimental design is considering the effect of a mutation, such as knock-in, knockout, transgene, at any stage in the mouse life cycle, and the strain is on a congenic background, you could choose wild-type or heterozygous litter mates or colony mates as controls. Additionally, you could choose an inbred mouse that represents a very similar background. But what if you're using a strain that has a mixed background? Wild-type or heterozygous litter or colony mates are going to be the most accurate representation of the strain background. In some cases, you may be able to use an F2 hybrid as well. Typically, F2 hybrid strains are used when there are two background strains present in your mutant strain. F1 hybrids are heterozygous at every loci for the parental strains and are identical to one another. But these hybrids lack the segregation that is present in a mixed background. To account for that segregation, F2 hybrids are considered a good approximate control because they exhibit some of the diversity associated with the segregation of the genetic material from the two parental strains. In F2 hybrids, siblings are not identical to one another. If you have an experimental design that looks at the effect of treatment, surgery, or other intervention, it is common to use the same strain, but just have an untreated group. If you are studying the effect of a drug or therapy on mice after surgery, it may be helpful to also include a control without the surgery to control for any effect that intervention may have caused. You may also consider an untreated control group if you are comparing the effects of two therapies. There are other control choices that may be better suited to your experimental design. But as always, it's a great idea to first check the published literature for similar studies and view their rationales for choosing one control over another, as this can help you make a more informed control choice. It is not only important that you choose the inbred strain that matches your strain's genetic background, but you also want to make sure that you are choosing the correct substrain of that inbred line. This study from Birdie et al. Examined the effect of JNK2 on acetaminophen-induced liver injury using a JNK2 knockout on a C57BL6 background. The researchers assumed that the knockout strain was on a C57BL6J substrain background. When compared to the C57BL6J control, the JNK2 knockout appears to lead to an increase in liver damage. This for them was a very unexpected result, as the researchers thought the knockout would produce less liver injury than the control mice. As a result, this led the researchers to go back to the collaborators to double check if they had used the right substrain as control. However, they were in for a surprise. They found that these knockout mice were actually on a C57BL6MJ background. And of note, C57BL6J and C57BL6MJ are actually genetically different. When the researchers compared their JNK2 knockout data to C57BL6NJ, they found liver injury was higher in the B6NJ than in both the B6 and knockout mice. Originally, the data made it seem that having the JNK2 mutation could prevent liver damage, but when compared to the correct control, the conclusion changed to the mutation contributing to liver damage. This example highlights the importance of when selecting controls that you are choosing the correct substrain as well. In this case, littermate controls would have been a better choice, along with making certain what substrain of B6 they were working with. It is important to note that different strains that are housed through different vendors may also differ, even if they are referred as the same strain or same substrain. I really can't emphasize enough the importance of knowing the history of your strain. As shown by my last example, not only should you know the genetic background of your mice, but also know the substrain information as well. One of the most commonly used strains in research is the C57BL6, otherwise known as the B6 strain. There are over 40 different C57BL6 lineages, though, that have diverged from the original lines created here at JAX by Dr. Little. Many of the lines have been separated for decades and have spontaneous mutations that have become fixed in different lines over time, making them each very unique. However, when researchers are reporting the results in the literature that pertain to the B6 inbred strain or mutations that may have been on that genetic background, often they do not report or include the substrain information. In nearly half of the publications that cite C57BL6 in C57BL6 mice in a 2013 PubMed search, the substrate designation or the institution from where the mice were obtained was not provided. And that poses the hard question of how can you be sure that your results can be reproduced if you do not include the substrain or at least the origin of your mice? Again, I can't stress this enough, but always put the full strain name nomenclature and the vendor or institution from where you obtained the mice in your papers, as complete nomenclature really does benefit everyone. Now, for the final part of today's presentation, let's talk a little bit about how genetic drift in your colony can impact their use in your research. So, how do substrains or different inbred strains arise? Anytime that colonies have been separated multiple generations, there are random genetic changes that occur and can become fixed within those populations. This is also known as genetic drift. Genetic drift is typically thought of as a bad thing, but not all genetic drift is bad. It can lead to species diversity. It can also lead to mutants with phenotypes that resemble human disease phenotypes. For instance, here we see a mutant mouse that arose in a colony at Jax and has a mutation in laminin alpha-2 morosin, which, when homozygous, produces muscular dystrophy phenotype like that observed in patients with Mersin-deficient congenital muscular dystrophy type 1A. However, genetic drift can also lead to data diversity and data that is not reproducible over time, which understandably can be super stressful. So let's look at this example of drift leading to a coat color change. All of these mice are C57 Black 6J. However, they each have had a change in their coat color due to mutations that spontaneously arose in genes that affect coat color. So, for example, on the top left, we have a AWJ mutation, which contributes to the agouti coat color. On the top right, we see a mutation on the tyrosinase gene, which contributes to the albino coat color. While you may assume that a change in coat color is not impactful, the mutation to the LIS gene that results in dark gray coat color also results in mice that exhibit immunodeficiency. The KIT mutation leads to mice that exhibit anemia and fertility issues. Most genetic drift is actually invisible, though, meaning that it does not produce a visible change in phenotype, such as behavior or physical trait differences. It was estimated actually by Russell and Russell that a phenotypic or visible mutation in mice occurs approximately every 1.8 generations. However, the rate of genetic drift is likely higher because it does not take into account invisible mutations. So it is important to understand that genetic drift is inevitable to any living colony, and there is not a way to completely eliminate genetic drift from occurring. However, there are some steps you can take to minimize genetic instability in your colonies and to make your research more reproducible. One way is to monitor your mice for phenotypic changes, maintaining pedigree lines and good record keeping, like a pedigree ledger, cage cards, and just documenting any noticeable changes. Avoiding selection pressure, choosing breeders randomly, and avoid using mice based on things like body weight, mice that produce the largest litters, or mice that are the easiest to catch, as doing so inadvertently is promoting genetic drift. To prevent your mice from becoming their own substrain due to genetic drift, we recommend refreshing colonies every 10 generations or so, by backcrossing them to their inbred background, through new breeders, obtained from the original vendor, or by using cryopreserved material from an earlier generation. Consider cryopreservation for colonies that you will not use in the next 6 to 12 months. Cryopreservation is really the closest you can come to truly stopping genetic drift. In addition, you can always recover cryopreserved mice from earlier generations if undesirable mutations develop in later generations. Cryopreservation has the added benefit of providing insurance as well in case of a catastrophe or, let's say, a lab contamination. So, what do you do if you see evidence of drift in your colony? Let's say we start with initial founder pairs, then breed them to get 2 litters of mice. Then we establish sibling meetings from litter 1 and litter 2. With this strategy, you will now have 2 pedigrees, and you never want to interbreed them between each other. And if you continue to breed this way, you essentially have 2 different colonies. The reason why this is a recommended strategy to prevent genetic drift is because if you have an unwanted mutation that arises, let's say a coat color allele that you can readily see, you want to shut this whole pedigree down, and you can use the other pedigree that is unaffected. You might be thinking at this point, well, if we know that these two mice are affected, which here I've shown in red, can't we just put them aside and no longer use them for breeding and just continue to use that green pedigree? In this case, even if this is one where we can see the mutation, it makes us highly suspicious that other mice in the same pedigree may carry the mutation. Because you have pedigreed lines, it's less likely that the mutation will spread through the entire colony and can avoid having a spontaneous mutation from affecting your research or halting it altogether. Of note, however, is that if you maintain a sister-brother breeding scheme long enough, let's say 20 generations, you will generate your own substrain. You can prevent this substrain development and decrease the accumulation of spontaneous mutations by refreshing your colony after 10 generations or so. Now, if you do find yourself in the position where you have only maintained a single pedigree, have maintained your colony for many generations without refreshing, or otherwise suspect that your colony may be genetically different from the parental colony, these are all important things to consider when you're choosing the appropriate control. As I discussed earlier, choosing the correct control in any experimental design is very important. So if your colony is genetically different than the parental colony or has drifted from an inbred background, you will likely need to consider wild type or littermates as control options. These will be the best representation of the genetics found in your experimental cohort. Now, I know I went through a lot in today's webinar, but I hope that I was able to provide a good overview of how to find a mouse model and some of the considerations you need to think about when using them for research. The Jackson Laboratory does have on-demand webinars that address each of these topics today in much further detail, but my goal was, again, to provide a general overview of all of these things. And thank you again for listening and joining me in this presentation.